science, physics, and biology. And I really liked how she was able to make the topic interesting. The things that the activities she had us do. So I started considering that maybe I wanted to be a teacher just like her. And then as I moved to high school, then I started watching a lot of documentaries. As I told you, I was really interested in field biology. And my actual dream was to develop a career as a marine biologist studying marine mammals in the ocean and studying bioacoustics, which is the science of communication. Basically, what people do in bioacoustics do in the wild, in the ocean, is they put microphones in the water and record sounds emitted by the marine mammal of interest. In my case, it would have been a humpback whale, and played back in other areas to other animals of the same species to figure out what kind of behavioral reactions you're inducing. And the goal is to try to understand the language that these animals employ to communicate with one another. Now, when it was time, so I spent a whole summer on a boat studying dolphins in the Mediterranean Sea for about 3 months. And I really, really enjoyed it. But it turned out that it was very difficult for somebody like me with no connections to develop a career like that. Now, mind you, I studied in Italy. I know here in the US, there's labs that do this kind of research that are well funded. In Italy, that's not the case. And so I had to let go of my plan A and consider my plan B, which was also something that didn't work out, but not because I couldn't get into a program, but because I wasn't good at it. So my plan B was to study molecular genetics, which was a very strong discipline in the university where I studied. And I realized that that wasn't really for me. So then I found an opening for a laboratory technician job in a neuroscience lab, which I hardly knew what it was. I had taken only one elective class in my 5 years of my biology degree. And it turned out that I really enjoyed it. It had a lot of things in common with the bioacoustics. I actually use electronic tools to measure the currents, tiny little currents that neurons use to communicate with one another. I can study a network of neurons communicating with one another, very similar to a pod of whales communicating with one another. And I can really try to get to fundamental questions. My main interest has always been learning and the role of experience in shaping the brain. And so I could really get to that quite deeply. So after a couple of failed attempts, you see, I found the right path for me. So now, once I figured this out, the path to getting to where I am now is very long. And you can see here the evolution of the career. Let me see if I can find the laser pointer. So this is Liceo Scientifico, is actually the science high school, which is where I began getting interested in biology. And then, so that was 5 years. It's 1 year longer than your high school. And then I got a degree in biology, which is a mixture of a bachelor degree and a master degree. It's a 5 years degree at the University of Pavia in Italy. And I also did my PhD in the same university. But now, after this was after a year off between undergraduate and graduate school, in which I tried the molecular genetics lab, and then I became a technician in neuroscience. And I finally entered a PhD program in biophysics. Biophysics, because neuroscience takes advantage of physics approaches and math approaches to biology. And it's a very quantitative discipline that studies
Oh,
excitable
excuse
membranes.
me? I'm sorry
Yes,
about that. I think your slide thing isn't moving. For I
the
yeah.
slides are moving. Oh, that's interesting.
Yeah,
Let me
I think
see.
there's something off about that. I'm not sure. It might just be like a tech issue.
Let me see. Let me stop sharing and let me just share the whole thing.
I know it
Oh,
sometimes happens in Google Meets. It's like really weird. Like it's very on and off.
let me see if sharing my entire screen. Okay, entire screen. Let's see. And now I can call up my presentation. And I was on this slide. Can you see it?
Yeah, I think we can see it now. Yeah, looks great.
So you should be seeing an image of a lady going from baby to senior. Is that what you see?
Yeah, that's exactly
Okay.
it, isn't it?
So now you should hopefully. Now it's not going to let me do the slideshow. Let me see if I can get it to work. Aha. I think we got it, right?
Yeah.
So you tell me if it moves. I'll tell you when I'm moving it. So this slide was just to show you the progression of what I had to do to be where I am right now, which is a full professor at the university. So I was telling you there's the Liceo Scientifico, the high school, which is 5 years, and then the biology bachelor and master degree, which is a 5 years degree. So this is technically a college degree. Is just that for Italians, it was longer at the time, and in the US is recognized as a bachelor and master, whereas in Italy it would have been just a bachelor degree. And then the PhD in biophysics, which was 3 years. And then at this point, I really wanted to pursue a career in neuroscience. I was ready also to move away from the country that was needed. And I found myself a postdoctoral training position at Brandeis University in the Boston area. So I was a postdoc for 6.5 years. Uh, Basically, a postdoc position is a more independent version of a PhD, meaning that at this point you have reached your highest education degree, but you are learning the ropes of how to manage your own lab in the future. So it's a lot more independent. You're expected to develop projects and run projects by yourself and begin to mentor other people. So from postdoc, I became an assistant professor at Stony Brook University in 2008. And I received my promotion to associate professor and tenure 5 years later. And then another 5 years as associate professor, and I became full professor 2 years ago. So this is basically the highest level of the academic career. Besides from that, there's only recognitions like distinguished professor, or so these are more like award situations, not so much career positions. So as an assistant professor, associate professor, and full professor, I do the same thing, which means I teach undergraduate students and graduate students, and I run my research lab, write projects. The only difference between assistant and associate is that my job becomes permanent. So in this transition, basically, I become a fixed employee of the university, and hopefully, if everything went well, it means that I have a lot of resources to run my research, and students and postdocs who do experiments, and we design projects together. So now I told you how I got here, but what I wanted to tell you is also what I do. So I mentioned at the beginning that I'm interested. Did you see the slide moving?
Yeah.
Okay. So I mentioned that I'm interested in learning and how experience is influencing neural circuits. I began doing this work as a PhD student, but I was working in a different system. And then as a postdoc, I was working on another system in the visual cortex. Initially, I was working on the cerebellum. And now that I have my own lab, I actually work on the taste system. And the reason why I got to taste is because it's basically one of the senses that all animals have in common. We all need nourishment to grow, but we also enjoy food and enjoy taste. So the part that I'm interested is really understanding the circuits that are involved in creating the sensation of taste and the sensation of pleasure associated with taste. So what the taste system does is very interesting because it transforms chemicals into sensations. And this begins at the tongue, but is really coming into the hemispheres of the brain in an area of the brain that is called the gustatory cortex and is on the side of your head. And here, information about who the taste is, so its identity, whether it is sucrose, a sugar, or quinine, a bitter, or sodium chloride is a salt. MSG is the taste of protein, is monosodium glutamate, and is the taste of amino acids. And then citric acid is the sour taste that is typically associated with fruit. So sour can be very sour when like a lemon or fruit that is not ready to be eaten, or can be subtle and pleasant like in strawberries that are ripe and ready to be eaten. So how is our brain making this information available to us to make decisions about what to eat and what to avoid? And then how are our preferences for taste developing from a young age to adulthood? So these are the type of questions that I'm after in my lab. So taste, you're usually thinking about taste as something that happens in the tongue, and it begins in the tongue indeed. So on the tongue, we have what are known as taste buds, which have many different shapes. So this one at the tip of the tongue are the fungiform taste papillae that contain taste buds. So the taste bud is that little round thing that you see at the top of this hole. And the papillae are these elevated or groovy structures that are full of taste buds. So the fungiform papilla exposes this bump to the taste that is coming in. The foliate papillae basically create these grooves so that the chemicals can sit in the groove a little bit longer and become in touch with the taste buds. And the circumvallate papillae, which are the ones that we have in the back of the mouth, are even wider and fuller of taste buds so that you can really have a full exposure of the taste receptor cells to the taste. So a taste bud contains a lot of taste receptor cells. This TRC is a taste receptor cell. And on top of the taste receptor cell, there's epithelium of the tongue with a little bit of a hole, the taste pore, so that the molecule can come in and activate one or more of the taste receptor cells. I color coded them here because taste receptor cells tend to respond to one category of taste. So say cells that respond to sweet stimuli, in this case, will be all blue, and then the salt will be all green. and the bitter would be the red, for example, right? They're obviously not these colors in your mouth, but I just color coded here to show you that there's some very specific taste receptor cells. So once these are activated, taste receptor cells begin to communicate with the brain through the endings, the nerve endings of the cranial nerves. We have 3 cranial nerves coming into the face, and some branches of them come into the tongue. I'll show you a little bit later what they look like, but what I need to tell you is that we don't only perceive sweet, salty, umami, which is the taste of proteins, or bitter. We actually, in the mouth, through all of the receptors that we can have sensations for many other qualities, including temperature. That really changes the taste. For example, think about a warm apple cider versus a cold apple cider. It's a very different sensation, right? We can also detect a spiciness, menthol, fats. We have receptors for fats in the mouth. We can detect different textures, different levels of astringency, carbonation, crunchiness. So you can see that it's not just those very simple 5 qualities that we have here, but each of these quality is also perceived a little bit differently depending on these other qualities that we can perceive. And so here is the circuit that is involved. So I mentioned the tongue quite in detail. The tongue contains the taste receptor cells contact the cranial nerves, the glossopharyngeal, the hypoglossus, and the vagus nerve. And they all branch out in areas of what is called the brainstem, which is this part of the brain that sits under the hemispheres and is like a bit of a tube. From here, so we have the tongue, we have the brainstem, and from the brainstem, information about taste comes into the gustatory thalamus. The thalamus is a region under the hemispheres that you're usually associating with what the brain is, and is basically a gateway of sensations. The thalamus has many different sections. There's a gustatory section, there's a visual section, there's a somatosensory touch section, hearing. The only thing that the thalamus doesn't have is smell. And then what the thalamus does is it sends that information to the appropriate cortical area. In the case of taste, to the gustatory cortex, which is on the side of the brain. I was telling you about the fact that taste also has a pleasure component. A lot of the information about pleasure is because the thalamus also contacts a region that is called the amygdala. And this region also communicates with the gustatory cortex, but basically receives the same taste information and is involved in adding content about the emotional state of the subject or the pleasure associated with the food. Like when you eat chocolate, for example, if you like chocolate, chocolate is sweet, but is also making you feel very good, right? And so it's part of the experience of taste to bring together all of these factors. So one of the things that gets attached to and helps create what is a food preference is the hedonic value of a food. Hedonic value means whether it tastes good or it tastes bad. Hedonic is just a general word to say that it has a pleasurable or unpleasurable or aversive effect on you. In taste, this is pretty fundamental to be able to associate the right hedonic value with a taste, because it is an important thing to make good choices versus really bad choices, right? Eating spoiled food or detergents. So what is good for you is usually pleasant, and it contains what is called palatable food, stuff that you need to grow your body and maintain your body. It's usually salts, because you need electrolytes. If you lose too many electrolytes, your blood pressure drops too much. Sweet contains calories. Calories are the building block and the energy molecules that you need. And then umami is predictive of amino acids, which is also needed to build muscles, to maintain all sorts of functions, including brain function, because a lot of proteins are actually receptors that signal in the brain. And then stuff that is bad for you are usually bitters, because bitters, for the most part, are poisons. And real sours, like not the sour of ripe strawberries, but sour like spoiled food, or acidic stuff, which can also cause some harmful reactions. Now, by experience, you know that while bitter is aversive in most cases, you can learn to like some bitter things. Like a lot of us drink coffee. Coffee is certainly not sweet, but it is a form of bitter that we learn it's safe and maybe even pleasurable as we experience it. So palatability can be changed by experience and by learning. But instinctively, all of bitters are poisonous, and we would just stay away from them. One interesting thing about bitters is that there's a lot of bitter substances in green vegetables. And this is one of the reasons why many children, when they're very young, do not like their veggies, the dark green veggies, because they have a little bit of bitters in them, and bitter is instinctively aversive. You just have to learn that it's safe, it's actually good for you because it has a lot of vitamins and minerals, and then you learn to eat it. So one of the big questions that we ask in my lab is whether the experience with food at an early age influences food choices in adults, right? It is a very important question, in my opinion, because we all intuitively know that this is the case, but it turns out it's not really being studied. And it has a lot of ramifications, right? If the experience you had early in life with food influences food choices, maybe we can develop better nutritional designs for children to limit obesity issues or other forms of eating disorders later in life, or promote healthy habits. In addition, there's another category of infants that is at risk for eating disorders of some kind, but also for changes in brain development. These are premature infants that are born and are not able to eat from the mouth like a normal infant would be. They have to be gut fed because they have not developed the sucking reflex just yet. They're too young. And one of the questions that people ask often is the lack of sensory experience around the orofacial area, so the mouth and the sense of smell. Does that influence brain development? There's some hints in the literature that it might, and some of the work that we're doing in my lab that we're actually trying to get published right now shows that definitely brain development is influenced by the sensory experience of taste. So now this is a big question, right? But I work with animal models, and I told you that I am a very basic scientist. I don't work with people, with human subjects. So how do we address a question like this in a lab using the laboratory mouse? Well, one of the great things about taste, and one of the reasons why I decided to study taste, is that the positive and negative reactions to taste are very similar in humans and even in mice. So you can see here, there's 3 images of the facial expressions of a laboratory mouse, a primate, and a baby and an infant, to sweet tastings. And you see that all of them have happy faces, they lick their lips, they protrude their tongue, and they really enjoy what they're putting in the mouth. On the other hand, they all have also the same reactions to bitters. They open their mouth, they try to get whatever was put in their mouth out as soon as possible. Primates have gag reflexes, so they can also throw up if they want to. The laboratory mouse doesn't, so it's going to try to open the mouth as wide as possible to let the taste drip out. So then we have another problem, right? We've seen that the laboratory mouse is a good model because it has the same reactions as primates and humans. But then how do we transform the taste experience in a human into something that we can do in the lab and control, so we know what tastings are being experienced and what they're composed of. So in my lab, we developed a taste experience paradigm that takes advantage of 4 basic tastings. One is sugar dissolved in water at different concentrations. One is salt dissolved in water. Salt at a low concentration is very pleasant. At a high concentration becomes unpleasant. So in this case, we want everything to be pleasant, so we keep it fairly low. Same with citric acid. We don't use the citric acid at the level of the lemon. We use it more at the level of the ripe orange or strawberry. And then we use Ensure, which is a nutritional milkshake that I'm sure you've seen in supermarkets. And the reason why we added the Ensure is because it has a smell. It smells like vanilla in our experiment, the one we use in our experiments. It has fats, it has proteins, vitamins, and minerals, in addition to having some salt and some simple sugars and carbohydrates. So it's a complex variety in and of itself. So then the thing that we can do is we can give the tastings in the water bottle to the mice for a certain number of days. And then we can ask the mouse several weeks later to report to us what it prefers. Okay, so I don't know that I can run this video now. Let me see. Okay. So what you see here is a mouse that is approaching a small slit, like a port, this shutter door, which opens. And then in the back of the chamber, there's this moving table that is controlled by a computer. And these are bottles that contain either water or different concentrations of sugar in water. So the mouse will experience one bottle for about 10 seconds, and the port closes, the rack moves in a pseudo-random fashion, and a new bottle is presented, the port opens, and the animal starts again. So by doing this, we can build what is called a sucrose or a sweet preference curve. So what this is on the x-axis, we plot the concentrations of sucrose that we have dissolved in our water bottles. And then on the y-axis, we measure the ratio of the number of licks that the animal does to the taste, so to the sugar solution versus to water. So we can always compare. So the dotted line that you see here is the licks to water. And then the data points that you see is how the number of licks to the sugar solution changes as we increase the concentration of sugar. So the fact that it's so high here means compared to water, it means that the animal likes this sugar solution much better. Mice have a very pronounced sweet tooth. So this is called a psychometric sucrose curve. We can do it for animals in different age groups. This P means postnatal, and 35 means 35 days. And naive means that this is an animal that has never been exposed to any other tastings aside from their regular laboratory food, which tends to be fairly salty. So we do these experiments at 35, 56, 70, and 91 days. And we see that at every age, 91 days is a full adult, 56 is a young adult, 35 is a juvenile mouse. So we can see that all of them at any age prefer sugar over water. Now, to calculate the licks, we have a little circuit that is activated every time the tongue of the mouse touches the zipper of the bottle. So we have very precise timing of the licking and counts. So with this psychometric curve, what we see is that there's no age-dependent changes in sucrose preference. At any age, the sucrose is preferred over water. So now what we do is we take our mice, and at postnatal day 22, which is when they're weaned, meaning that they begin to eat independently from their mom, we give them 2 cycles of the 4 tastings that I showed you before. So on each day, we change their water bottle with a bottle with a sugar solution. This is now 200 millimolar, so it's about this concentration. They really like it a lot better than water. Then we give them citric acid in the water bottle. We give them Ensure and sodium, and we repeat it twice. So it's an 8 days exposure paradigm. And then we put the animals back on their laboratory food for 4 weeks. And then we go get their psychometric curve 4 weeks later. Why did we do that? Because we wanted to ask what is the long-lasting effect of such an 8 days exposure. We didn't want to see what happens right after. So this is just to show you again what we did. And now I can show you what happens to the sucrose curve. So this low one is the same as the one you saw before in animals that were naive. So they only had the laboratory diet. And the ones that had those 8 days of exposure at the time of weaning, now they really, really, really like the sugar a lot more. You can see here that they lick a lot more to it, and they drink a lot more of it. And the star here means that this is statistically significant. So then we had to learn a little bit about what is making this curve shift. So what I showed you here is that the taste exposure at weaning modulates the preference for sweet. This is not depending on the fact that sucrose is also one of our stimuli, because if we do 8 days of sucrose instead of all the variety, the curve doesn't shift. We know that we need calories, because if we modify our exposure paradigm to substitute sugar with saccharin and ensure with MSG, which is the taste of proteins, but both of them have no calories, we don't see any shift in preference. So we know we need the variety because a single tastant doesn't do anything. And we also need smell, because if we make the animals anosmic, meaning that they can't smell during the time in which they're exposed to the tastant, even if their smell recovers soon after that, the curve still doesn't shift. So we have a combination of factors of the taste experience in the mouth and in the gut that modifies preference. And then the other thing that we do, because I'm a neuroscientist and I want to look into the brain, is we try to go see if something in the way neurons respond to taste has changed in animals that received that exposure. So here what we do is we have a mouse that has a little window for us to look into the taste cortex on the side behind the ear. And the mouse is sitting under a microscope. It's alive. It's fine. Is trained to sit still and to lick a spout that is moved in front of their face. They do 5 dry licks, meaning that they lick the spout 5 times before a drop of water or different concentration of sugar are delivered. Okay, we repeated this thing many times. So this is time. This is the number of times we repeated it. And you see for every trial, there's 5 dry licks, and then the drop arrives, and these are the licks to the drop. Okay, and we can do it many times. And then what we can do here, this is a very cool experiment. So we implant a prism on the side of the head because microscopes have a beam of light that is vertical, but we need to image on the side. So on this side of this triangle of this prism, there's a metal surface. So the laser beam that we use to look at the surface of the brain, to illuminate the surface of the brain, bends on this metal, on this mirror side, and bends 90 degrees so that we can deliver it to the area that we want. And so by doing that, we can measure activity. So here, this dark thing, so this is the surface of the brain that we see from the side. This dark thing is a blood vessel that we use as a reference point. We know that the taste cortex is around this section of the blood vessel. And the white speckles that you see, those are neurons. We make them express a gene that lights up when certain substances are flowing into the cell, in this case, calcium ions. And so when we do the experiment, we can see the movie. You see now that some cells light up, right? A lot of them actually light up when sugar arrives. So we can then use this sequence of images to figure out and time it to the licking to figure out who was activated at what time and what is the way they respond to the concentrations of sucrose. So to summarize very quickly some of the things that we learned is we learned that taste experience changes how neurons in the taste cortex are activated when the animals licks for sucrose. They are actually much better at discriminating one concentration from the other. And then what we did do is we asked if we take the same exposure paradigm, and instead of starting it at the time of weaning, we started in adult animals, would we see the same shift in preference? And we don't. So what this tells us is that the experience that is needed to really shape preference in adulthood is the early experience. is the one that a subject has very early in life when they begin to eat independently. And then through a series of other experiments, we also learned that taste experience influences the development and maturation of brain circuits. So it is a fundamental experience, and without it, the development of the brain either slows down or remains behind. So this is what I wanted to tell you about my science. And then I had some prompts for the talk. So there were some requests about what my daily duties are. My daily duties over my career have changed a lot. So my daily task and duties as a PhD student and postdoc was to run the actual experiment, execute it, obtain the data, analyze them. And this is sort of the system that I use. There's a little chamber with a piece of brain, and we use these special electrodes that allow you to record single neurons one by one. You can record the current that is flowing in their membrane, and you can quantify their action potential. And action potential is a very powerful electrical current that allows a neuron to communicate with another. Or I did experiments like the one I showed you with an animal under the microscope doing a task and imaging its brain. Now, my tasks are mostly to write projects, design projects, discuss projects with my trainee, apply for grant funding to be able to pay the stipend of my students and postdocs and the resources my lab needs. So it's more like an entrepreneurial job. I run a small company now, practically. So I am responsible for making sure that my research is well funded. I have the resources that I need, and or that everybody in the lab needs to do the experiments that we need. And then I'm responsible ultimately for what comes out of my lab. So manuscripts for publications. I provide a lot of feedback to my students and postdocs, and I am responsible for the data that go out. So I am responsible to make sure that we present stuff that is truthful in a rigorous fashion. I teach undergraduate students and graduate students, so I am sometimes in the classroom. College classrooms can be very large. At a public institution like the one where I work, my undergraduate classroom has about 120 students. Graduate classrooms are much smaller. They're usually about 8. And then my job is to make sure that I can communicate my science like I'm doing today with you, but then also to other scientists, possibly to funders. I mentor my students and postdocs and help them develop their career. I try to inspire people to be excited about the science. And of course, I have to do all of this in a very rigorous way so that the scientific enterprise maintains trust in the people who fund it. Because a lot of the funding that I obtain is from the National Institute of Health, which distributes taxpayer money to research. So I have a responsibility toward taxpayers to be a good scientist and provide reliable knowledge. So what are the pros and cons of my job? Well, there's many pros and I really enjoy it. The biggest pros of all is that now, especially now that I run the lab, but also when I was a trainee, I get to choose projects and ideas that I want to develop. As a trainee, in cooperation with my mentor and advisors, because I am learning to run a project on my own, and now together with my trainees, because it's part of their career and it's part of my lab productivity. I get to collaborate with really smart people, develop new ideas, thinking ways that I had never thought before. And sometimes you find something that is really exciting, opens a new avenue of research. It's really something that you want to develop further. There are also cons in science, right? Sometimes an experiment doesn't work. So a failed experiment can be painful and set you back quite a bit in time. However, a failed experiment is also telling you that your way of thinking was not right in terms of biology. And so rethinking it and reorienting the work may actually benefit from this. Obtaining funding and publishing scientific papers can be challenging. So you go through rounds of revision. You have sometimes that a project gets rejected. So you have to learn how to read comments from your peers, get better at asking questions, become more rigorous at running your research plan. And then this is a pro and a con at the same time. So my job has flexible hours. Flexible hours meaning that even when I was a trainee, I was the one who set the schedule based on the type of experiments that I needed to do, and I would set my own schedule. I do it now too, and now that I write most of the time, I can really do it from anywhere. So the negative part of the flexible schedule in a job like mine is that when you have an analysis of data ongoing and when you have a project that is going, sometimes you just don't stop. So having flexible hours means just working a lot. But you have the opportunity to set your own schedule, which is something that many jobs don't really have. And so this is all I wanted to show you for today. And I'm ready for your questions if you have any, about the career, about the science, anything.
Thank you so much for the presentation. I really enjoyed it, and I think I definitely want to look more into neuroscience. So, one question I have for you is like, what is your favorite part of your job? Like, if you could pick anything that you do nowadays.
So that I do now, what I really, really enjoy is to interact with young people who come in the lab with a lot of enthusiasm and ideas, and we get to develop them together, and I see them grow into scientists, and then, they go on to develop their own. When I was younger, the most fun I had was actually running the experiment, and sometimes I still miss it a bit. I
Oh,
see a
okay,
raised
great.
hand.
Mhm.
So I was wondering if you had any like student internship opportunities at Stony Brook for anyone who is like interested in this topic.
Yes, I sometimes have high schoolers in my lab. It's not super easy for a student in high school to run a project in a lab like mine, because as you probably know, you're not allowed to work with animals. There's limitations in what you can do, and we are not allowed to have any tissue that is selectively for a project for a high school student. And so we are limited in what you can do. But I've had 5 high schoolers coming through my lab over the summer, over the years. It depends on the project that is ongoing at the moment, and if I have a good mentor that can follow the student, yes.
So
Okay,
I was
thank you.
also wondering, aside from like programs provided by like schools, do you have any tips for like how to reach out or find research opportunities?
At this stage, so at your career stage in high school, your best bet is to reach out directly to professors, but expect to have to reach out to many of them. My advice is that because it I can see it with me. Not every summer I have a person that is at the right career stage that can mentor a student, because maybe I have very junior trainees in the lab, so I can't associate you with a very junior trainee because I'm still helping them learn stuff. Sometimes I have a graduate student who's writing their thesis and getting ready to graduate, so they're busy with that, right? So it has to be the right time, and it would be the same for a different lab. However, if you contact someone, um, make sure in your email that you indicate specifically why you're interested in what that lab does, which means take a few minutes, look at the webpage of the scientist that you would like to work with. You don't necessarily have to read one of their scientific paper, because sometimes they can be really difficult. But having seen the website, having seen what kind of questions they ask, makes for a better informed email. Sometimes you get a very generic invitation email, like students interested in doing research, and that's all they say to you. It's great, because I know that as a high schooler, that's already quite advanced to even consider going to a university lab to do research. At the same time, if you don't tell me why, I would probably advise you to go somewhere else. The reason is the research that I do in the lab is not very friendly for high schoolers. So I know it. You probably don't, right? It's not friendly in terms of requirements and training. So I need somebody to be at least interested in the types of questions that we want to ask. And then we can design a little project that is age appropriate.
Okay, so I think if everyone's good with questions, thank you so much for being here today and presenting all this information to us. It was really interesting, and I'm like really looking forward to researching more about like the career of neuroscience and also just like laboratory research in general. So we will get back to you on more information. Shreya can continue like talking about some stuff.
Yeah, thank you so much for this presentation. Like, I didn't even know taste was something that you could research. So this like really opened my mind to new ideas of things that I can research about. So thank you so much for this.
You're very welcome. So I'm gonna let you go enjoy your Sunday.
Thank you. Thanks everyone for coming, and thank you especially to Dr. Maffei.
Bye-bye.
Bye.
Thank you. Bye.
Thank you. Thank you.